We're going live.
Good evening, everyone, members and guests, to the second lecture of December 2020 of the Astronomical Society of Glasgow. Tonight, we have an observers meeting telling you what's in the sky tonight with Dr. Andrew Conway, who's going to take the first 10 minutes of tonight's lecture time. And then we will move on to the main item, where we will ask Dr. Rosa Santomartino to tell us about I like this, wee little creatures in space. So without further ado, Dr. Andrew Conway, can I ask you to tell us what's in the sky tonight?
Okay, thank you very much, Craig. So I'll just share my screen. Right, can I just check, can everyone see my screen, which should be a sort of twilight, star-filled night sky? Can people see that? Okay, yeah?
Yes.
Good. So, what this is going to be mainly for those who are not so used to looking at the night sky. So if you know your way around the night sky, then apologies, you might know a lot of this, but it won't last for long. So what we're looking at here is the night sky as it would have looked this evening at about 5 o'clock. And the tree line you see here is from Mugdock Country Park, just above the visitor centre, and the rise above the visitor centre, which is where the ASG is going to put its observatory, the Eric Tomney Memorial Observatory. Now, part of the reason I was interested in this is because the first thing that you might want to be looking at if we do get a clear night sky in coming days is you'll be wanting to look low to the southwest of the sky. So you see here, this is southwest here, south is here, and you'll see something not too unremarkable. Very low down, you'll see a waxing crescent moon. But it's these two things, which you might at first mistake for a pair of bright stars next to each other, very bright stars, in fact. But you'll maybe notice they don't twinkle as stars do, because they're not stars. They are the planets Jupiter and Saturn. And what's remarkable about them is they're, as you can see, very close to each other in the sky. And on Monday, the 21st of December, they will be about the closest they've been, I think, if I understand correctly, since the 13th century. And they only get this close every 20 years. And this is a particularly close, what we call conjunction between these two planets. So don't worry if it's cloudy on Monday night, because you know any of these nights, coming nights, they will be very close together, but they'll be at their closest on Monday. So we get two planets for the price of one here. The only slight snag is they're very low down. So as you can see here, they're about to sink beneath the trees. So if you've got trees or a building to your southwest, you might want to go upstairs or maybe walk off down the road a wee bit to get a better view. Now, those aren't the only planets in our evening sky. If I pan round over here to the southeast, we have much higher in the sky, we have, and still very bright, is the planet Mars, which you will see as, it's called the red planet. It doesn't look red as such, because your eyes are not very good at distinguishing colours in low light levels, but it certainly does look reddish. It will look yellowy, orange, definitely. You might notice it says here, I don't know if you can make out, that says Uranus is there. You won't be seeing that with the naked eye. Rarely some people can, but with a pair of binoculars, and if you look at star charts, you might be able to find it. But yeah, Mars, again, very easy. You'll see it with the naked eye, no problem whatsoever. Now, what I want to really talk about tonight is the winter constellations. Now, it just occurred to me earlier today when I was thinking about this, that it jogged my memory of an evening when I was 4, maybe at the most 5 years old. I remember coming back from some Christmas party, going into the back garden of my parents' house, and it just snowed. It was a cold, crisp, clear December night, absolutely gorgeous night. And snow had settled along the washing line in my parents' back garden. And as a little boy, I would jump up and ping it, and it would create a mini snow shower. And after all those little snow flakes finished twinkling down, I noticed that they weren't the only things twinkling in the sky. And I think that is my first memory of seeing the winter night sky. I didn't know what the constellations were back then. That's probably my first memory of noticing that not only are there stars, but they're very beautiful. And they're particularly beautiful in winter, because there's some very impressive constellations to see. So what I'm going to do is start the Earth turning. And as I speed time up, you'll notice that the sky is changing because the stars are rising. And it's not the stars that are moving, of course, it's that the Earth is turning. So if I pan round to the southern part of the sky, you'll see that this bright group of stars here, which is, of course, you've probably heard of it, it's the constellation of Orion. Many of you will recognize it. I'm going to stop it right there at midnight. So this is, to my mind, the classic winter sky set of constellations centered around Orion. Now, the downside is at the moment you have to wait till midnight to get this view. It means Orion rises a little bit earlier than that, but to get this view that I'm showing you, you'd have to wait till midnight. But because not only is the Earth turning, but because the Earth is going around the sun, the stars and the sun rise 4 minutes earlier every day, in a sense, not the sun, but the stars. And that means that after you've waited a month, what you see here will occur 2 hours earlier. So you'd see this at 10 o'clock in mid-January. In mid-February, it would be 4 hours early. You'd see this at 8 o'clock. So if this is past your bedtime, wait up till midnight to see this. The good news in astronomy is if you just wait a few weeks, wait a month, you can see it at a much more civilized time of the evening. So that's the constellation of Orion there. It's notable because there are two very bright stars. There's the bright star Betelgeuse, which is in its shoulder, it's sort of an orangey red color. And there's the bright star in its foot called Rigel. And the third brightest star is of note to Harry Potter fans because it's called Bellatrix. And these three stars here were also made famous in a film, Men in Black, many years ago. It's Orion's Belt. I think Orion's Belt's more famous than that film now, though for a while it was the other way around. And if you trace these three stars down, you arrive at Sirius. And this is called the dog star because it's in the constellation of Canis Major, which is a dog. And there's a little puppy up here, Canis Minor, the little dog, with the bright star Procyon in it. So I'll turn that off because it's a little bit distracting. Now, Orion, as you might have just seen there, is actually fighting Taurus the Bull. Taurus the Bull is up here. And this orangey star here, about nearly as bright as Betelgeuse, but not quite, is Aldebaran, the eye of the bull. And if you keep going, you'll get to the Pleiades, also known as the Seven Sisters. And that's a splendid sight through binoculars. You can see it with the naked eye, but even through a modest pair of binoculars, you'll be starting to see something like this. You'll see there's actually more than seven sisters there. If you see seven sisters with the naked eye, you're doing really well. If, like me, you can barely make out one sister with your glasses off, then you need an eye test, definitely. But if you've got a good telescope and some imaging equipment, and Colin Robbins' imaging group could certainly produce images to equal, if not better, this, then you'll make out the fact there's actually some what's called nebulosity. There's some gas that I think it's actually not in amongst the stars of that cluster, but that certainly is a young bunch of stars that have formed comparatively recently, much younger than our sun. They're youngsters by astronomy standards, at only tens to hundreds of millions of years old. It's not very old in astronomy terms. Now, the final thing I'm going to show you in this very brief tour of the night sky is actually just underneath Orion's belt. It's sometimes referred to as a sword, and sometimes this is referred to as the gem in the hilt of the sword. And you'll see it with the naked eye. You can just about see it in the light polluted suburbs of Glasgow. But again, it's something that binoculars will improve greatly, and it's the Great Orion Nebula. And you see it here on the screen there. You should be able to see, with a small telescope, you'll see a lot of this structure. Again, this is a star-forming region, younger than the Pleiades. But as I say, something you can see without binoculars, without a telescope, with the naked eye, is a little smudgy patch below Orion's belt. Now, that is just a little taster of what you can see in the night sky. If you'd like to know more, then you can use the same software I'm using right now. It's called Stellarium. It's free and open source software. You can get it for any kind of computer. I've also got it on my phone. I think I have to pay a few pounds, which I think goes to support the developer, so I'm more than happy to do that. There are plenty of other options as well, but Stellarium is the one that I would recommend. Also, the Astronomical Society of Glasgow run an excellent beginners class. It's run by Lorna Napier and Chris O'Kane, and I recommend that if this has whetted your appetite or you just want to know more and you're just starting out, please do go along to the beginners class. And of course, do consider joining the society if you've not already done so. So I hope that gives you a very quick introduction as to what you can see in the night sky. Thanks very much.
Thank you, Andrew. I never get fed up listening to the way of listening to someone describing the night sky. Although I've known it for over 50 years, it's still amazing to me when I look up. I still have that boyhood, interest in the sky. Thank you anyway, Andrew. And we want to move on to the main feature of the evening, which I would like to introduce to you, Dr. Rosa Santomartino, who's come over from the University of Edinburgh. I'll just give you the synopsis that was in the newsletter, and it's about microbes, the space microbes, the importance of studying wee little creatures in space. Microbes are everywhere in the Earth's biosphere, including the human body, and will necessarily follow us during space exploration. As they play many important roles on Earth, they will be crucial in our survival in space. In this lecture, Dr. Santomartino will explain why it is important to study microbes' behavior in space and how we could harness their power to explain what is discovered on this matter with the Biorock experiment launched aboard the International Space Station in 2019. Rosa Santomartino is a researcher in space microbiology and microbial astrobiology at the University of Edinburgh. She has a BSc and an MSc in biotechnology and a doctorate in molecular biology. During her research, she has worked in the UK, Italy, France, USA, and Mexico. She performed 2 space microbiology experiments, one launched in the International Space Station in 2019, and here's where we got an update, and one ready to be launched at the end of 2020. I will leave Dr. Rosa to tell you because that one has already been launched, and I'm sure she will update us on the progress of that particular mission. So without further ado, Dr. Rosa, I give you the stage.
Thank you so much also for this kind invitation. I'll try to share my screen. If I manage to do that. Right, sorry, I don't know why. I think I have to move. Okay, found it. And also share my audio. Okay, right. Do you see all the screen correctly?
It looks good.
Great. Okay, so thank you for this invitation. I'm very excited of being here. Actually, the first time that I got the invitation, it was in July, I think. So such a long time ago. I'm very happy that I'm finally here. So thank you for giving me this opportunity. And so today I'm going to talk about space microbes. That are basically is the science of space microbiology, and I hope that I will convince you about the importance of studying microbiology and microbes in general in space, because they are going to be quite important for us out there, and I hope again that I will convince you about that. So what am I going to talk about today in this talk? First of all, I'm going to define what I mean with we. So in other terms, what is a microorganism? Then which are the roles of microorganisms on Earth and also beyond Earth, so the possible roles of microorganisms. Which are the conditions that are present in space? So you are probably more familiar than me with all the space conditions because basically my background is in microbiology, but I'm going to give you some insight into the conditions in space from a biological perspective. And then I'm going to give you some examples of some space microbiology experiments that have been performed. Actually, I choose 4 of them, 4 key ones. And then I'm going to talk about the experiment that I performed luckily together with my supervisor, Professor Coquel, and a wide group of researchers last year on the International Space Station. There is the Biorock experiment. And so we're going to talk about biomining in space, and then something about what to expect from the future. So first of all, what is a microorganism? This here is what we call evolutionary tree of life. We biologists call it evolutionary tree of life. This is, of course, just a drawing representation. And this is basically a graphic representation of all the organisms. Basically, it allows us to classify all the living organisms, all the non-living organisms. And as you can see, each branch basically represent a different type, a different group of organisms. For instance, here we have the branch of plants, here we have the branch of animals, and we are here. And then there are others. So when we talk about microorganisms, we are actually referring to this type of group. So the group of archaea, the group of fungi, particularly unicellular fungi, the group of protists, and the group of bacteria. So as you can see here, they are all quite diverse. Why are they called microorganisms? Because of course they are very small. In fact, micro comes from in the order of microns. That is 1 micron is basically 0.001 millimeter. So very tiny organisms. To see them, you normally need at least a microscope, and they can be of different types. So they can be unicellular algae, they can be archaea, bacteria, unicellular fungi. They can also be viruses, although it's kind of controversial if viruses are really alive or not. So in other terms, are they really living organisms or not? And they can be also protozoan, like for instance, amoeba. So as you can see here, they are all quite diverse. However, they have some characteristics in common. The first characteristic that they have in common is that they are all made of a single cell, also called unicellular organisms then, or even less. In fact, as I mentioned before, viruses are composed of even less of a cell. And another characteristic that they have in common is that they are everywhere on Earth's biosphere. This is a direct consequence of this in the sense that being made of a single cell, they are quite simple. And the fact that they are simple make them also quite adaptable to different conditions. And this is why they have been found almost everywhere on Earth's biosphere. And indeed, when I mean everywhere, I literally mean everywhere. In fact, they have been found on soils, and they are actually quite important to make the soil fertile. They have been found on infrastructures, pipes, and you can see here that it's quite dangerous when this happens the majority of times because it causes a lot of damages. But they have also found on very hot environment, very cold environment, as well as very dry environments. Very salty environments. This is an image from the Dead Sea, and it's actually Dead Sea. The name comes from the fact that they thought it was a dead sea, literally, so that nothing could grow there. And actually, they found out that some microorganisms are able to grow in these very hot, very salty conditions. So Dead Sea is not so dead after all. They have also been found on clouds. And last but not least, they are also present on plants as well as animals, including our own human body. And in fact, that's just a curiosity. It has been estimated that for every single human cell that composes our body, there is one microbial cell in our body. It means that basically the ratio between our real human cells and bacterial cell or microbial cell is 1:1. And although this may sound scary, this is actually a very good news for us because the microbes that compose our body, or actually that are present in our body, are absolutely essential for our own health. It's called the microbiome. Maybe you heard about this word. Microbiome is basically just the ensemble of all the microorganisms present in a certain environment. And this environment can also be our own gut, for instance. So when we hear about microorganisms, we normally think about something that is nasty. We normally think about something nasty, and it's particularly true in this time in which we are in a pandemic, of course, but more often they are actually quite useful for us, not only useful, but necessary for our own survival and for our own flourishment. So in order to try and convince you about this, I'm going to now shortly summarize which are the roles, some of the roles that microorganisms perform for us on Earth. And for instance, they are absolutely important in the food industry. One for all for whiskey. Without microorganisms, we won't have whiskey, for instance. They are also important for the oxygen present in our atmosphere. In fact, while we hear a lot about the importance of trees and plants for the oxygen, when in early Earth, the composition of the atmosphere was quite different. And at a certain point, a number of a certain type of microorganisms called cyanobacteria are bacteria that can do photosynthesis, as plants basically, started producing oxygen in such an extent that they caused what is called the great oxidation event. And this is the event that basically caused the percentage of oxygen in our atmosphere that allows us to breathe nowadays. So basically, if we can breathe, and actually if we evolved, it's thanks to this event, and so thanks to some bacteria. They are also important in some bioindustries, for instance, biofuel, the production of biofuel, but also in the production of some biopolymers, like for instance, biocomposite, bio-cotton, and bio-wool. They are extremely important in the drug industry, in pharmaceutical industries. Just to make an example for all, they are basically the total quantity of insulin that is produced is produced by microorganisms, by some bacteria. They are also important in the recycling of waste. And one thing that has been recently discovered is that there are also microorganisms that can actually biodegrade plastics, so they can really eat anything. Again, from an environmental point of view, they can also perform a task called bioremediation, that is basically the use of microorganisms to clean some polluted environment from certain compounds. These I mentioned already, they can help us producing, they produce fertile soil. They make the soil fertile so that we can grow crops. And last but not least, they can also perform a task called biomining, that is basically the extraction of minerals using microorganisms, minerals and metals more in general. And I'm going to talk about this a little bit more in detail later. So I hopefully convinced you that they are extremely important for us on Earth. So imagine how important they will be for us beyond Earth, on an extraterrestrial planet. And some of the possible roles of microorganisms beyond Earth are, for instance, in the generation of oxygen. We saw how important they were on early Earth, so we expect them to be quite important also on another planetary body. Again, they are going to be important for the production of fertile soil from extraterrestrial regolith and extraterrestrial rocks. And this is strictly related to the fact that the microorganisms could help us for the generation of food, because if we have fertile soil, we can create crops, and this comes automatically with the generation of food. They are also going to be important, again, for the recycling of waste. So a space station, this is quite an interesting point of view, because a space station, as well as an early extraterrestrial settlement, is going to be necessarily a closed environment, an environment that cannot have much exchange with the external environment, actually. So the recycling of all everything that we can, as much as we can, is going to be quite important and quite relevant in an enclosed environment such that. And last but not least, in a future in which we are going to be completely settled in space, then we need to advance our civilization there. And so we are going to need also bioindustries, and microorganisms, as we saw, are quite good from this point of view. So all these possible tasks that I explained here hold the final aim to going and support what we call self-sustaining life support system, and also to support what we call in situ resource utilization, or ISRU. That is basically just the concept of relying, of basically use the resources in the place in which we are going to settle, rather than constantly resupplying from Earth. That is not sustainable, as you can imagine, but the farthest we will go from Earth, the less this will become unsustainable. So we need to rely on resources in the place in which we are going to be. Okay, I told you why microorganisms are important. I told you that they can grow basically everywhere. But when we think about a harsh environment, in my opinion, the harshest environment that I can think about is, of course, space. And I'm going to explain you which are the conditions in space. First of all, I'm going to talk about the condition in open space. And I'm going to talk about how these conditions affects actually biology, so living organisms, particularly microorganisms. The first condition that we will find in open space is, of course, vacuum. Vacuum is a condition of absence of matter, or also extreme low pressure. Normally, when we are in low Earth orbit, that for those that are also called LEO, that for those of you that are not familiar with these terms, it basically means it's the orbit in which the majority of space stations like the Mir, the International Space Station, and also the space shuttles orbits or used to orbit. In this condition here, in this orbit here, vacuum goes from 10 to the power of -7 Pascal to 10 to the power of -4 Pascal, so very, very low. In this condition, a living organism will experience two main type of problems. Let's call them like that. First is desiccation. So basically, the water will evaporate from the body or from the cell surface, and so we are going to desiccate, literally, or at least our external surface. That when you think about a microorganism that is made of a single cell, that can be quite a problem. And the second is the asphyxiation, because of the fact that being surrounded by vacuum, there is also an absence of oxygen. So we are going to asphyxiate. However, please forget these type of sci-fi images in which you see that an astronaut is going to explode, the eyes are going to blow, etc. This is not going to happen, but you are still going to die in a matter of seconds or minutes, so you're not going to be very happy anyway. Another extreme condition that you will find in space is the extreme temperatures. For instance, in deep space, the temperatures goes between 2 Kelvin and 10 billion Kelvin, so quite extreme. Again, when we are in low Earth orbit, the temperature actually depends on the position of the spacecraft or your position, basically, in respect to the sun, as it's clear from this image here. So when you are closer to the sun, of course, you will have the highest temperature, that is around 120 Celsius degrees. And when you are farthest from the sun, you're going to be at -152 Celsius degrees. You will have extremes in these two type of temperatures in around 90 minutes. That is the time of a typical LEO orbit. And so they are quite extreme as well. So when they build a space station, a spacecraft that is going to be in LEO, they have to survive and tolerate this type of temperatures. Another condition in space is, of course, radiations. This is probably one of the most common ones that you can hear. The radiation that are present in open space are cosmic radiation, galactic and solar cosmic radiation, the Van Allen belt radiation belts, and also the solar electromagnetic radiation, for instance, the UV radiations. Why are they so bad for a biological system, for an organism? Basically, because the high energy that is that composes the radiation itself can seriously damage all the molecules that composes an organism, particularly the DNA. So this damage can be direct or indirect. Either way, the damage can be irreversible. That can lead to both, to either cell death or also mutation that can lead to even worse scenarios, like, for instance, cancer, if you are a complex human being, of course, or an animal. The Fourth condition that we can find in open space is microgravity, so basically the low gravity condition. On Earth, the gravity acceleration at the surface is 9.81 meters per second squares, of course, also called 1 per g. When we are in condition of low or orbit, but this is true actually for everywhere in space, the force of gravity is not actually removed. However, an object that is orbiting in LEO experiences a constant state of gravity-induced free fall, because it is orbiting basically around the Earth. And this is what we refer to when we talk about microgravity, that is most commonly called the weightlessness or something like that. Microgravity is normally, it's the condition in which gravity is less than 10 to the power of minus 2 g, but is more typically around 10 to the power of minus 5, minus 6 g, so quite low, quite close to zero, but not zero. I'm not going to talk about the effects of microgravity on animals and plants, because that's such a big topic that can take a whole lecture. But I want to briefly introduce you to the effects of microgravity on microorganisms. So when it actually is quite controversial in the scientific community what is actually the effect of microgravity on the smallest microorganisms, because it has, in order to directly sense microgravity, a cell needs to be larger than 10 micrometers in diameter, and the majority of bacteria and yeast, fungi, and so unicellular fungi and so on are smaller than this. So it means that actually the majority of microorganisms cannot really sense directly microgravity, but what they can do sense is the indirect effects of microgravity on them. And I hope this is quite clear from this image, I'm not sure, but when you are, this is a cell basically, when you are on Earth, gravity allows some mixing, some natural mixing of nutrients. So basically the nutrients, not naturally, but like the nutrients can mix and arrive close to the cell and be incorporated into the cell. And on the other hand, the waste material that comes from the cell go away from the cell. But when we are in microgravity, for a series of physical reasons, basically there is no mixing anymore. So nutrients cannot easily reach the cell, and equally the toxic waste products of the cell cannot easily go away from the cell. So what a microbial cell senses in microgravity is that it's intoxicated microenvironment surrounding basically the cell. Now, I spoke about the condition in open space, but when we are inside a spacecraft, like for instance an International Space Station, the conditions are different. The majority of the space experiment that I'm going to mention in a minute have been performed inside a space station or a spacecraft. In that condition, they won't experience vacuum or extreme temperatures, but they will still experience some type of radiations, like for instance the cosmic radiation, as well as microgravity. So this is something to keep in mind. Another topic is also which are the conditions that are present on planetary bodies. And here I will focus only on two main planetary bodies, because these are the two that are most, let's say, famous for when we talk about the human space exploration. There are Moon and Mars. So let's see which are the conditions that some of the conditions that we will find there. On the Moon, vacuum is still going to be present because the atmosphere is considered negligible. And for the same reason, the temperatures are quite extreme, so there are extreme oscillations. Radiation are going to be present as well, and they are going to be roughly similar to interplanetary space. So we are also going to have this condition. We are not going to have microgravity, but we are still going to have a low gravity that is 0.16 g. And we have to add another term here, that is because we are on a planetary body, we are also going to have rock, or in other words, regolith. The regolith on Moon is poor in nutrients, and it is also the dust that composes the regolith is also going to be quite toxic. So if we want to go there and create a space settlement, you have to keep these in mind as well. What about Mars? Let's see. So from the point of view of vacuum, Mars has actually an atmosphere, but it's quite toxic as well because it's high, basically because it's high in carbon dioxide, so CO2, but not just that. And also, yes, there is an atmosphere, but it's quite a light atmosphere. So there is no vacuum, but the pressure is quite low. So it's still going to be a problem. From the point of view of temperatures, yes, the temperatures are quite cold actually, between -87 and -5 Celsius degrees, but probably not limiting, particularly for some type of organisms. So this is a little better. Then from the point of view of radiation, we are still going to have some radiation, like for instance, the galactic cosmic rays and solar energetic particles. Again, we don't have microgravity. We have a low gravity that is higher than lunar gravity, but is still lower than Earth gravity. That is 0.38 G on Mars. And then from the point of view of regolith or rock, it's quite toxic, unfortunately, for living organisms because it's rich in chlorine and related compounds. So these are the conditions that are on these two type of planetary bodies. Okay, now I told you a lot so far. I hope I'm not boring you. Let's go on and try to understand why do we want to study microorganisms in space. So now you know a little bit about the background, but you can still ask, okay, but why do we want to study microbes in space? Why is it relevant? I still don't get it. Well, I can split this question in two. The first can be why microorganism, and the second, why in space. And these two questions are actually quite linked. For instance, why microorganism? Well, because we saw that they can survive to the harshest environment. And this is quite relevant for space because we saw how extreme the conditions in space are. So studying microorganisms in space can actually help us understanding which are the boundaries of life. And this is the main topic of astrobiology, for instance. Then they are also important. There are also important implications in human health, and from the space perspective, this is going to affect the microbiome of astronauts, so the collection of microorganisms on the space crew. They are important in bioindustries, and they are also essential to create an habitable extraterrestrial environment, for instance, the formation of fertile soil. And these are all going to be important for the human space exploration and for an extraterrestrial settlement. They are also quite cheaper as organisms in respect to other type of organism that you can study. Not necessarily true, but in general, it's quite true. And also, just for the sake of knowledge, just to know what happens to microorganisms in space, why not? But there is also another good reason to do that, and it's this one. As I mentioned, they are everywhere. And when I say everywhere, I also mean our own body. So it doesn't matter where we will go in space. We can go wherever. We are going to take our microorganisms with us. So it is better to study them, also to try and understand how they behave in space in order to use them as to understand how they could be helpful and not dangerous for us. Okay, let's go a little bit more now, not into details, but I would say more. I'm going to give you some example of some space microbiology experiments that, in my opinion, are quite remarkable and explains quite well why space microbiology matters and can be useful to study microbes in space. The first one is this experiment that was conducted on a space shuttle in 1996. This experiment was quite important to understand the importance of the ozone layer for life on Earth. So what did they do? Basically, they have taken one bacterium called the Bacillus subtilis that you may have never heard about it, but this bacterium is extremely widely used in space experiment because it's a very tough one and it can resist high dose of radiation, a lot of very harsh environments. So it's a very good model system for space experiments. And they used this microorganism here, this bacterium, they put it on basically a surface and they create a kind of biological dosimeter for radiation. And they then exposed this biological dosimeter to the UV radiation, actually to solar light solar radiation through basically through a screen that was simulating the ozone layer, and there were different screens with different thickness in order to simulate thicker or thinner ozone layers. The authors say that this is quite, the results were quite interesting because it was like the demonstration of the value of space experiment as a time machine, as they call it. In which sense time machine? Because with this experiment, they could predict the sensitivity of life to a shrinking ozone layer and also study early biospheres. So go kind of back in time and try to understand what happens to the early organisms that were evolved before the ozone layer was built up on our Earth. The second experiment that I want to show you here is kind of related to the first, but not completely. It was performed on the Russian Mir space station in 1997. And it was important to understand the role of vitamin D for skeletal muscle tissues in space. I don't know if you know about this, but astronauts normally experience a loss of bone density during space flight, and it's around 1% of loss of bone density every month that they spend in space. So with this experiment, they wanted to understand that by exposing the astronauts to UV radiation, they could produce enough vitamin D to counter-interact this negative effect on bone density, basically. So what did they do? They created a similar dosimeter using, again, the bacterium Bacillus subtilis, and they asked the astronaut to sunbath, literally to sunbath, to a quartz window that was on the Mir station, wearing this biological dosimeter. Then they analyzed the samples, and the results were quite interesting because they discovered that indeed sunbathing could produce enough vitamin D to counter-interact the loss of bone density. On the other hand, however, it was quite dangerous because the number of the dose of radiation that they were accumulating by sunbathing was too high, so they actually recommended the use of dietary supplements instead of sunbathing. This experiment here, it's an experiment that I want to mention because it was from 2006, as you can see, and was from a space shuttle mission. This was the first demonstration of something that is widely established now in the scientific community, and it's the fact that the microbes normally express a higher virulence when they are in space. So they are more virulent when they are in space. This was the first demonstration of that. And basically they demonstrated that then by studying Salmonella typhimurium, that is a pathogen agent that you may, well, Salmonella I think is quite common as a disease, unfortunately. They grew them on board the space shuttle, and they discovered that the virulence after the space flight was increased by 3 to 7-fold, so quite a higher an increase in virulence for these microorganisms. That's quite scary if you think about the fact that together with this increased virulence of microbes and bacteria in space, astronauts in space also usually experience a lower immune system. So the two things together can be quite scary, and this is also why it's important to study microorganisms in space. Also, it poses the base for to conduct research to use spaceflight and microgravity in particular to develop vaccine and antibiotics. The last experiment that that is not mine, that I want to talk about is quite recent. It comes from 2019, and basically they analyzed the microbial community of on board the International Space Station. So as I mentioned before, the space station, as every spacecraft, is necessarily a closed environment. The space station in particular has been active for 22 years now. And it's now, unfortunately, showing some corrosion and damage. And some of this corrosion that you can see here is caused by some microorganisms, such as mold, for instance, and bacteria. So what did they do with this experiment? They collected samples from 8 different places on the International Space Station, and they just analyzed the microorganisms that were present in these 8 places. What they discover is that the microbiome, so the collection, the whole collection of microorganisms that they found around the International Space Station resembles that of animal skin surfaces rather than, for instance, the microbiome that is present in the clean room in which they assemble the ISS. And although this may sound a weird result, that's actually quite obvious if you think about the fact that for 22 years, almost the only organisms that were that went into the International Space Station are humans. And so that's quite clear why they have basically all the microorganisms that are, or almost all the microorganisms that are around the space station, inside of course, not outside, are associated with humans, with human microbiome. And some of them, scary enough, are opportunistic pathogen. Opportunistic pathogen are some microorganisms that are not normally dangerous, but they can become dangerous when you have a low immune system, for instance. Now, let's go back a little bit. I told you a little bit about some roles of microorganisms on Earth, and I mentioned very briefly this possible role here that is this role here that is biomining. What is biomining? Biomining, that is also called bioleaching, these two terms are actually used as synonyms, is the process of using microorganisms, microbes basically, to extract useful elements from the rocks or also from mine waste. It's also used for environmental issues, like for instance in the process of bioremediation that I mentioned before. And not everyone knows that actually biomining on Earth is currently used to extract around 20-25% of copper, 5% of gold, but also nickel, rare earth elements, uranium, zinc, and other metals. So it's widely used on Earth. And there are some advantages in respect to traditional mining. For instance, it's quite complementary to more traditional methods, and it can also improve the rates of metal recovery from mine waste, for instance. But it's also generally more economic and generally also more environmental friendly. So there are a lot of advantages for using biomining. There is an increasing interest, though, on the process of biomining in space. So this is not a reality yet, but a lot of people are thinking that biomining in space could be quite a good thing to do. Well, it's clear to understand why, if you remember about what I told you about the in situ resource utilization, that was that concept that said that it's better to use resources in the place in which we are going to settle rather than resupplying constantly from Earth. So biomining in space can be really important from this point of view. And when people, researchers in particular, think about, but also industries, think about biomining in space, they are particularly interested in understanding if it's possible to biomine Moon, Mars, and asteroids. And I want to say also that this type biomining in space is thought to have, of course, got a lot of advantages from the point of view of space settlement. So it's going to benefit space settlement. But there are also researchers thinking that it could benefit somehow terrestrial industries. This is probably not sustainable at the current state, but maybe in future, who knows? So it is with this idea in mind, so trying to understand the feasibility of space biomining, that our group first proposed this experiment called Biorock. This experiment was actually proposed for the first time by my supervisor at the University of Edinburgh, Professor Cockell, Professor Charles Cockell, maybe some of you know. He's quite an important astrobiologist. And he first proposed the Biorock experiment in 2008 at the ESA, so European Space Agency Geobiology for Space Exploration and Settlement Topical Team, or GISSE. However, as you can see here, this experiment was funded by STFC. It was supported by European Space Agency, and you can see here all the logos of the different, representing basically all the different researchers and students that were involved in the project, and without without them, it wouldn't have been possible. So I have to thank them first of all. Which was the motivation for Biorock? Well, of course, we wanted to understand if biomining of extraterrestrial material in space was possible, as well as using extraterrestrial regolith for both in situ resource utilization and also for supporting life support systems. But before understanding if these things here were feasible, we first needed to understand which was the effect of gravity on all these processes. In other words, how does gravity, the low gravity that is commonly present in space, how is it going to influence the way which microbes interact with the rock surface, the mineral surface, or the surface in general that we want to biomine? Now I will show you this video that was prepared by NASA in preparation for Biorock. Biorock was actually launched and completed on the International Space Station in 2019, summer 2019. So we already have the results. But this video was prepared in preparation for Biorock, actually. So you will hear that all the verbs are with future tense. But I still want to show you this because I think it's an excellent video and explains very well the concept behind Biorock. So please raise a hand or unmute yourself if you cannot hear the audio of this.
The International Space Station will soon host some of the smallest miners in the universe, microbes. Microbes growing on the surface of rocks can gradually break them down and extract useful minerals and metals. This is a process called biomining. As we explore space, we are seeking to use biomining to turn rock and regolith into soil for growing plants and food. But before we can use this technique in planetary settlements, we first need to test it in space. On the space station, bioreactors will be placed inside a centrifuge where microbes will grow on rocks in microgravity and simulated Martian gravity. Investigators will examine how 3 types of microbes behave within pieces of basalt and evaluate how well the different microbes extract elements from the rocks. The findings will be compared to ground-based results. We hope to gain insights into how microbes interact with rocks in microgravity and how we might use them in our exploration of deep space.
Right. I hope you could hear, but nobody unmuted, so I will assume it was okay. So let's get a little bit into the details of that. The concept was very well explained by this video. I really liked that video, I must say. But now let's get into the details again. So first of all, we needed to select the right microorganisms for the experiment. And the right microorganism needed to have some characteristics in common. For instance, it had to come from natural rock or soil environment. It needed to have possibly a demonstrated capacity to extract elements from the rock. If possible, needed to be desiccating resistant because of how desiccation resistant because of how the samples were prepared for the launch. Possibly again, they needed to have been previously used in a space experiment, easily studied in the laboratory, and also, last but not least, they also needed to be safe for the space crew. So eventually, we decided to go for these 3 microorganisms. The first is a bacterium called Sphingomonas dissecabilis that was provided by our group at the University of Edinburgh. The second was Bacillus subtilis, that should sound familiar now because it's the same that was used for the other 2 experiments that I showed you before. And it's, as I said, it's quite a good organism for space experiments. So we decided to use this, and this was provided by the German Aerospace Center, or DLR. And the third was Cupriavidus metallidurans, provided by the SEK Centrum in Research Center in in Belgium. And this organism here is quite, has been isolated basically from environment polluted by heavy metals. So we know that it could have been a good microorganism to study as well. And then we needed to select the natural rock substrate. As the video mentioned, we we chose basalt for two main reasons. The first, that basalt is a generic porous rock material that means that it could provide a good support for the bacterial colonization of the surface of the rock. And also, basalt is actually a good analog for moon and Martian rocks. So we selected a specimen coming from Iceland, and we cut it into basalt slides that were 13, 15 millimeters wide and high and quite thin. So we called it basalt slide basically. And then we prepared our sample. So how did you do that? Well, basically we prepared a single bacterial culture, coming from a single microorganism, and a single species of bacterium were inoculated into a single basalt slide, basically. So we had several basalt slides, each one containing a different type of microorganism. And also we had some sterile microorganisms, some sterile basalt slides, meaning that they were basalt slides without any bacteria inoculated on that. These sterile basalt slides composed what we call non-biological control or abiological control, or also negative controls. So they were just controls, basically. Then, after preparing all the samples, they were integrated into the hardware that is called the Biomining reactor, or BMR. And this hardware was specifically designed by Kaiser Italia and by our group for Biorock, for the Biorock experiment. As you can see here, each hardware can allocate 2 different basalt slides. You can see here an empty one. And this is just to show you what was the environment inside each of these culture chamber, basically. So each square here is basically a little chamber in which that was composed basically by the basalt slide with the bacterium integrated in that, and then also by the solution containing the nutrients that were allowing the bacteria to grow. These are, again, the hardware inside the external container. So in this case, you can see some example of hardware that were ready for the launch. And then finally, this is us in July 2019 at NASA Kennedy Space Center in Florida. You can see here me in both pictures, very happy. This is Professor Charles Coquel, and this is Annemiek Waken. She's a PhD student that collaborated with us for Biorock. And here you can see charts that is very, here you can see us very happy because we finally finished preparing all the samples and they were ready for the launch. So here you can see charts handling the samples to the International Space Station processing facility, ready for the launch again. And here you can see part of the Biorock group. This is me here, me here, and me here. Enjoying basically some tour around the Kennedy Space Center after hard work. And then the rocket launch that hosted our experiment was, the mission was SpaceX Commercial Resupply Service 18. You can see, this was the rocket launch here, and this is me on the launch site. So that was quite exciting because I could really attend the rocket launch. This is the Dragon capsule when reaching the ISS. And here you can see the astronaut Luca Parmitano that is installing the hardware into the cubic incubator on board the ISS. Cubic is just a type of incubator that has this centrifuge in the middle, but they also have some spaces around available. And these are European Space Agency facility that was used for this experiment. Here you can see again Luca Parmitano performing some images, taking some images of the hardwares, and he took these images just before and just after the end of the experiment itself. And it turns out that actually these images, despite just being pictures, were actually quite informative, because allows us to show, to observe some features that were not observable anymore when we had our samples back on Earth. These features were, for instance, the aggregates that are indicated by these yellow arrows here, and also these gas inclusion here that are pointed out by these blue arrows here. Again, this was quite interesting because when we had our samples back in our hands, these features were not visible anymore, meaning that if we didn't have this image acquisition session, we never knew that this was happening. Then, after approximately 1 month of being in space, they actually reached us back on Earth, and here it's Charles and I, very relieved that everything was okay and that we had our samples back in our hands. And this is me analyzing the samples. Right, enough with pictures of me and other people around. Let's get a little bit more into the details of this experiment. So we prepared 4 separate sets of samples, each one containing one of the 3, or actually all the 3 organisms, as well as a biological control in triplicate. So we prepare 4 sets of samples that were identical one to each other, each one having all the set of samples again. And the 3 of them were launched on the International Space Station, and they were subjected to either microgravity, simulated Martian gravity, so 0.38 G, actually was 0.4 because of the instrument, the incubator that we used, and simulated terrestrial gravity. So we also have a simulation of terrestrial gravity on board in space. That's quite a rare opportunity. But we also had a 4th set of sample that was subjected to real, of course, it was on ground. It's what we call ground control. And in this case, the samples were actually subjected to real terrestrial gravity, so 1G. As I mentioned before, that the experiment was, the samples were on the ISS for approximately 1 month. However, bacterial growth, so the real experiment, actually occurred for 21 days. Okay, so I told you that this was launched last year, so it's been more than 1 year now. What did we discover? Which are the results that I can show you about this? Well, the first result that we got from the Biorock experiment regard the bacterial growth, so the final cell concentration, final concentration of the cells that we got. And turns out that all the samples that were in space, regardless of the gravity conditions, they all reached the same final cell concentration after 21 days of growth. What does this mean? Well, from a biological perspective, what is the meaning of that? Well, because of how the experiment was designed, we don't know what happens during the experiment. Unfortunately, we only have results from what happens after the end of the experiment, so at the 21st days. What does it mean? It means that we don't know, again, what happens before. We don't know if the growth went like this or like this. We have no idea about that. However, the fact that we got the same results, the same final cell concentration after 21 days, regardless of the gravity condition, is a very good news from the point of view of biotechnological applications in space, particularly on Mars, on the Moon, or other planetary bodies that have a low gravity condition in respect to Earth gravity, because it means that gravity won't negatively affect bacterial growth, and so eventually, probably, hopefully, biotechnological applications. So this is the first thing, the first result that we got. Low gravity is not a limit to biotechnological applications in space, at least in principle, of course. But what about biomining? I made a big deal about biomining, space biomining. So what did we have from this point of view? In order to address this question, I first have to introduce you to the rare earth elements. I don't know if you ever heard about this type of metals. These are metals. This is, maybe I should mention, this is periodic table, chemical periodic tables. Rare earth elements are the elements that are underlined here in yellow or orange. And these are elements that, despite the name, are actually quite abundant on Earth. However, they have an enormous impact on our everyday life because they are present in every single simplest electronic device that we use. So considering how dependent we are today on electronic devices, imagine how dependent we will be on extraterrestrial space settlement. We will probably rely on electronic devices even more. Where are found? When can we find rare earth elements? Well, on Earth, of course, as I just mentioned, but they are also present on some type of rocks on the Moon called CREEP, that stands for potassium, rare earth elements, and phosphorus. They have also been found on asteroids, as well as on some Martian meteorites. So we know that it's probably also present on Mars. They are also present on Mars. So what did we discover? We discovered that one of the three bacteria that we used, particularly Sphingomonas dissecabilis, is actually able to biomine rare earth elements in space in all the 4 gravity conditions that we tested. So that's a very good news. We also tested, and this is a very fresh data actually, we also tested another element. There is called vanadium. Honestly, I never knew nothing in particular about vanadium before observing these results, but now I made my research and I discovered that vanadium is such an interesting element, actually, such an interesting metal, because it has a strong, there is a strong industrial interest on this metal. Is widely used in alloys to enhance basically the hardness, the tensile strength, and also the resistance of the metal. And also is quite widely used in structural materials. So considering the harsh environment, the harsh condition that we are going to have on other planetary bodies, and I'm particularly thinking about moon dust particularly, vanadium may be quite an interesting element to address from the point of view of space biomining. So we tested also this, and we found out that 2 microorganisms, 2 bacteria that we used in the Biorock were actually able to extract vanadium in space, particularly Sphingomonas dissecabilis and again Bacillus subtilis. So these are very good news. The fact that we discovered that 2 bacteria were able to extract useful elements from the rock, regardless of the gravity condition, means that space biomining is in principle achievable under a range of different gravity conditions. And this makes also Biorock actually the first experiment to study space biomining on a space station. So likely a quite remarkable result. To summarize the results from Biorock, then, as I just said, we first demonstrated biomining on a space station, and we also demonstrated that this is in principle achievable under a different range of gravity regimens. Also, we also showed that final bacterial population are not affected by gravity condition after 21 days of growth, and this is hopefully a good news from the point of view of space bioindustrial applications. We also demonstrated the functionality of a novel hardware, the space biomining reactor, and we also discovered the novel biomining microorganisms, that is Sphingomonas dissecabilis, the one that was provided by our group here at the University of Edinburgh. So Sphingomonas dissecabilis was already discovered as a microorganism, but nobody knew that it could actually perform biomining. And that was quite an interesting result, specifically because from the point of view of rare earth elements, because there is a big interest in finding organisms that can extract rare earth elements, because as I mentioned before, they are quite important in our everyday life. So this is a demonstration actually that space microbiology can not only benefit space research, but also terrestrial application. Now, this was mentioned before during when you were presenting me. Because of the success of Biorock, we are now having a second space experiment that is currently on board the International Space Station. It was launched on the ISS when was it? 6th of December. So almost 2 weeks ago, basically. And the scheme, so it is currently there, is the experiment itself, the samples are there, the experiment itself is not started yet, it should start next week. However, this experiment is going to be quite similar to Biorock. We actually consider it a kind of follow-up of Biorock, but there are some key differences. The first difference is that we are going to biomine this type real extraterrestrial material, so we are going to biomine asteroid. And then the difference is also on the microorganisms that we are going to use, because this time we are going to use a bacterium again, our friend Sphingomonas distechabilis, that behaved so well in Biorock that we decided that he deserved a second ticket to space. But we are also going to use a different type of microorganism, that is a fungus called Penicillium simplicissimum. If this name sounds kind of familiar to you, it's because penicillium is the not specifically this one, but this type of microorganism are those that that produce penicillin, basically. So, and we are also going to use a combination of the two, because in nature microorganisms are normally together. You will never find a microorganism that is separated to other microorganisms that are isolated, or at least in the majority of times. So we wanted to understand if the combination of the two was going to create some competition or rather some synergy. And this time the group, we at the University of Edinburgh are going to be the main PI, the PI of the experiment. And this is going to be funded again by STFC and also supported by the UK Space Agency and also European Space Agency, actually. This is just to show you that it was successfully launched last Sunday. I should correct that. Sorry, it was not last Sunday, but the Sunday before, on the 6th of December. And here you can see the rocket launch. It was launched this time on the Commercial Resupply Service 21. And unfortunately, this time, because of the COVID situation, I could not go on the rocket launch and on the rocket launch site. And I had to watch it from my laptop, but it's still interesting to know that your samples are there. So I was still quite excited by this. So what can we expect from the future? There is a lot that is still need to be understood from the point of view of space microbiology. First of all, I hope that within, I don't know, 6 months or so, I'm going to give you results from bioasteroid. And I hope everything went okay and we are going to have good results from this point of view. But apart from this, from our own experiment, there is still a lot to know. For instance, we still have to understand better about the molecular, biochemical, and physical processes that are influenced by space flights. We need to understand more about the effects of gravity on microorganisms. And we also need to select the best microorganisms for a given application. We also need to understand how to scale up the system. So, okay, we show that space biomining is feasible, but if we really want to do that in an industrial way, we need to scale up the system. We only have a very small culture chamber, and so that's not going to be enough. There's not enough to say that is I mean, it needs to be scaled up. Then we also need to understand how long duration spaceflight are going to affect microbes, because one thing is being in space for 1 month, one thing is being in space for 6+ months, or also being there forever. And also, this is quite important to me, how to keep the process sustainable in general. And so research is needed from this point of view. And I want to stress again that this type of research not only benefits space science, but also terrestrial application, as demonstrated, for instance, by the discovery of the novel biomining microorganism that is Sphingomonas distechabilis. I want to conclude by thanking all the people that were involved in the Biorock experiment. Here you can see all the names of them, but they are never going to be inclusive of all the people that were actually involved in this. And I want conclude by leaving you these useful links if you want to know more about space microbiology in general. If you want to contact me, these are my contacts. This is my email, and I'm also on Twitter. And I will be very happy to answer your questions. And I thank you for your kind attention and again for the invitation.
I'll just say one word to you, Rosa. Fascinating. Absolutely fascinating. There's quite a few questions have flown in while you were going on there. I'm just trying to see if I can bring them up. There's some commentary, but there's also some I'm glad you've got a drink. You did well to get through all that. Let me just start at the top. If anyone wants to say anything, then kindly put your hand up and we'll let you talk to Rosa. Let's have a look. Right. The comment from Alistair says, sounds like the film Sunshine. I think that was to do with on the space station and the vitamin D. The vitamin D.
I didn't watch this movie.
Oh, right. Here's one coming in from Julian. If biomining is used on Mars for ISRU, will that not impact or prevent the search for life, exobiological life forms?
Yeah, that's a main issue when thinking about an extraterrestrial performing an extraterrestrial space settlement. This is something that needs to be kept in mind, but it depends on the. So let's talk about the future in which we are going to go on Mars and settle there. Then at that moment, there is nothing we can do. Our microorganisms are going to come with us necessarily. And so there are lots of planetary issues on that. Sorry, I forgot the name of that. Like, there are a lot of laws. Oh, planetary protection. That's a very common word that I usually use, but then I completely forget about that. Yeah, there are some laws that are the rules of planetary protection that says that nowadays you cannot actually go on, you cannot grow anything on Mars, you cannot send microorganisms of Mars, if I'm not wrong. So, but the problem is that if you really want to go on Mars and settle there, then I think you have to update planetary protection considering this in mind, because even if we are going, even a single person of us are going, we are still going to be an organism that are kind of contaminating the planet. So it really needs to be, this is something that we have to keep in mind constantly when thinking about that. And probably it's going to be updated.
So search for life before we go there.
Yeah, basically.
Thank you.
Thank you.
Okay, there's some commentary coming in from Robert Law saying the University of Dundee was involved with experiments with microorganisms in space, including worms. So rather larger than
nice.
Microworms.
More microworms. So you missed out the word micro there, Robert. Just to say that they had an exhibition in the observatory at Dundee, the Mills Observatory, including microscopes, so you could actually see them. Yeah. So there you go. So go to Dundee.
I didn't know about that.
There you go, a trip for Dundee. So just across the two bridges. There's a question from Frank Cohn. He says, hi, Rosa. You mentioned cyanobacteria being important for producing oxygen on Earth. We often regard cyanobacteria in the form of green and red blooms as being toxic. But can they help in removing carbon from the atmosphere?
So I'm not an expert in cyanobacteria, unfortunately, but when we have, what I recall is that when we have the blooming, it's because of basically an impairment of the equilibrium in the environment, like in the water that we have. So that's, if I don't want to say something that is not correct, but I kind of remember that there is a blooming sometimes when there is a pollution. I may not be right, so I will just drop it because I'm not sure. However, yes, they can be quite useful. And in fact, the majority of experiments that are aiming to develop basically those self-sustaining life support systems that I mentioned before have some compartments, are planning to have some compartments that have cyanobacteria, because cyanobacteria can be quite important also from this point of view. But as everything, as every organism, included ourselves, we can be either detrimental, problematic, or we can be very useful to something. And it's the same also for cyanobacteria. In some type of environment, in some condition, they can be quite dangerous, and in some other, they can be quite useful.
You can't tell one from the other.
Well, it's really that it depends on the it's a matter of ecosystem, you know. So it's not necessarily one way or another. For instance, I don't know, the opportunistic pathogens that I mentioned before. So those microorganisms that can be dangerous, that are normally quite okay. They won't hurt you if you are okay, but as long as you have a low immune system, they will attack you, and you will develop a disease. It's always an ecosystem, a matter of ecosystem and equilibrium. But yeah, they can definitely be useful, actually.
Hope you're not going anywhere. We've got 10 messages in here from your audience. Right, I've got one here. This is from David Dagan. He's obviously thinking about getting you back when you get the results back from back from Bioasteroid.
I hope I will have good results to present.
Have a live lecture, please. So he's obviously asking if you can, when you get your results and you get your mind clear, come back and talk to us.
Thank you. I would be very happy to do that.
Right. Oh, here's one from Roy Bryce. Bioasteroid. Am I correct in thinking that the basalt slides in the Biorock experiment were replaced by slides made on Earth from unpolluted inside of meteorites found on the Earth?
Yeah, kind of. So for Biorock, we used a basalt slide because I didn't show you the results from that because they are still ongoing. But one thing we also wanted to check with Biorock was also the biofilm formation. That is basically a little film that is formed by microbes on a surface, on a rock surface. So in that case, we used a slide. But normally when you perform biomining also on Earth, you crush the rocks. So this time for us, for Bioasteroid, we are going to use crushed minerals rather than what am I saying? Crushed meteorites rather than slides of meteorites. And yes, and so unpolluted. No, unfortunately, because they were not pristine, they were polluted, but we sterilized them before using them by basically baking them in an oven, in a very high temperature oven. And in this way, basically you sterilize it, you keep it, everything that was on it, it was sterilized, but unfortunately it was not pristine.
Right, okay. There's a question from Alistair. Will biomining for rare earth elements yield more common metals, etc., as a side effect, or must we mine these separately?
So no, actually, no. It depends, of course, on the several things, like for instance the metabolism of the microorganism that you are used, and yeah, it's basically, and also some other condition, actually environmental condition, I would say. But the biomining of, for instance, I spoke about rare earth element and vanadium. They happen together, so they are not necessarily separate. Of course, if you want to increase, let's say that you are interested in only rare earth elements, then maybe you want to focus your microbes to do only that. And in that case, maybe you can enhance you can teach your microbe with some molecular techniques by using one mineral rather than another and extracting that rather than the other. And in that case, you can probably enhance the extraction of some minerals and metals in respect to others, but no, they are not necessarily detrimental to others.
Thank you.
Thank you. Hang on a moment. Yes, here's one from Roy and Charlotte Evans. Hi, Rosa. Have billionaires like Elon Musk and Jeff Bezos been interested in this topic too? They might have the money to do bigger experiments and could find industrial uses, benefits from the results. I thought this myself. Can you guys get some money in there, because I think this could be profitable.
Yeah, well, the truth is that, so I don't know about them in particular, but there are industries that are trying to find a way to, for instance, collect asteroids for biomining in space, or like drag asteroids somewhere and then biomine, etc. But the reality is that so far the technologies are not there yet. So everything that they thought so far, from my knowledge, is not sustainable. So there are industries that are interested in that, but they still didn't know how to do that, how to really scale up the system. It's possible that we can understand how to scale up the system here on the ground. And actually, we are hopefully going to do that. I know our group in particular is interested in doing that, and we are also collaborating with some people in US, etc., to try and understand how to do that. But yeah, it's not a reality yet. With the Biorock, that's actually a good point, because I want to point out that with Biorock, we only were able to say, okay, in principle, this is achievable. But this type of experiment are just what we call basic research. That means that we can explain the process. We can explain that the process is feasible, but it's not an industrial application yet. There are some differences that you have to take in mind when you have industrial applications.
Thank you. Just it's a statement from Julian, who handles our website. He says he'll put your URLs and post them on the ESG website, so you might get someone coming back and talking to you.
Thank you.
Those will go on. Another one from David Dagan coming in. Are there plans to carry out this kind of research on the manned lunar missions planned for 2024 onwards?
Yeah, yeah, there are. I don't know exactly which one, but we are actually planning to do something of that. We know we are again collaborating with Luis Dea from University of Colorado to do something like that as well, and there are ideas of doing that. So that I still don't know if it's going to be a reality, if we are going to do that, but I know that there is a lot of interest from this point of view. So we are probably going to, we are hopefully, not probably, hopefully going to do something. And if not us, of course, someone else is going to do that.
Excellent. I have a question myself. I'm going back onto the human side of things. You mentioned that there was these bioorganisms are more virulent in space by 3 and 7 times. And you used the phrase opportunistic pathogens, which worries me slightly. If we're going to Mars and taking 6 months to get there, have the medical people got the answer to this, or are they working on it?
That's a big issue. In fact, one of the main problems when thinking about going to Mars, for instance, is that, as you said, you take 6 months to go there at least, and during these 6 months, you're going to accumulate a lot of radiations. That is already a problem, and then you're going to be there for a while, because you're not just going there for 6 months and then come back in 6 months. And then you are, so yeah, that's an issue, and we still don't know how to do that. So that's something that people that are interested in trying to understand how to get to Mars. And with Moon is a little different, is less problematic, because it is less days basically. So the radiation that you accumulate are way less than when you go farther, of course. But people that are interested in understanding if it's possible to go on Mars and also to settle there need necessarily to address these issues and this question. So no, we don't know now. Today we don't know how to do that.
I had another question. This is one of my advantage of being the master of ceremonies. I have another question. You had the 3 bio your 3 experiments in the space station, and you had another one on Earth.
Yes.
Does relativity come into that? I'm thinking of you know, the twin paradox, or is it too small? Do they not suffer from effects of relativity?
Well, I honestly cannot answer that, but what I can tell you is that from the point of view of final cell concentration, I didn't mention that because I thought it was too much into details and it was not necessary, but there are some differences between, so you saw that we had 2 terrestrial gravity samples. One was in real terrestrial gravity and it was the ground control, and one it was in simulated terrestrial gravity. So in theory, from the point of view of gravity, they were equal. But from the point of view of final cell concentration, we found some differences between the 2. And also when we compared, basically when we compared the space samples with the ground control, we sometimes found some differences that we didn't found between the space samples. So this means that gravity is not the only element that is affecting the result that we have, and it's actually quite good that we had the possibility to have both a terrestrial gravity control in space and on the ground, because this allows us to see that if we only had the space terrestrial, the terrestrial gravity sample in space or only the ground control, we probably didn't notice that. We didn't know that if we didn't have that. So
do we know why do we know why the differences are there?
No, unfortunately, we only have hypotheses. One hypothesis is the radiation that maybe have influenced that. But there are so many other differences, like the fact, for instance, that on Earth we had real terrestrial gravity, while in space the gravity was simulated by insertion into a centrifuge. So it was not, it was again a simulation, so that may also cause some problems. The fact that, yeah, it was a simulation rather than a real gravity. We don't know that, and it's very hard to rule these out because they were there are so many effects that could be there. Well, we only have hypotheses, I'm afraid.
Okay, I thought with setting it on Earth, you had something in mind with regards to the 1G on Earth and 1G in the ISS. But
one thing that you know for sure is that the only difference for the samples that were in space is the gravity condition. This is what you know for sure. But then for the ground control is much more complicated because, for instance, the samples that were in space were launched on a rocket. So there was some vibration that the ground control didn't experience, for instance. Or, well, apart from the radiation and the difference in simulation and real gravity, there are some, yeah, it's very different. It's very hard to rule out the different effects. While if you compare the samples in space, it's much easier because, as I say, they were subjected to exactly the same conditions except the gravity condition. So in that case, if you see an effect, you can really say, okay, that was gravity.
I thought you were going to say, look, it's all in space anyway, so we'll just count the ones in space. We know about the ones on Earth.
Well, but when you have this experiment, you really have to compare them. So,
Rosa, there's more questions. I've got one from Roy Bryce. Both experiments, would biomining at a large scale not be affected by the ambient heat as well? In the space lab, it is quite warm. In space, it isn't.
Yeah, well, if you want to perform it in space, you have to think about this as well. For instance, and it may also change depending on the microorganism that you use. So every microbe, but actually every organism has an optimum of growth at certain temperatures. So if you think that that is mandatory that they use that microbe because that microbe is going to be absolutely necessary for your application, then you need to stick with the temperatures that microbe requires. And in that case, you maybe want to use an incubator, for instance, or you have to create an incubator on Mars or whatever. On the other hand, if you think that the incubator is going to be limiting, then you have to find a microbe that perform what you want it to perform in the condition, for instance, in the temperature condition that you have. So that's why in the final slide, or the final couple of slides, I mentioned that you need to find the best microorganism for the application that you want to perform.
I think you have a lot of budding microbiologists here. Ed Tobias writes to you and says, fascinating talk, thank you. What is thought to cause the increased virulence of microorganisms in space? Is it the effect of the radiation upon the microbes? DNA? Question mark.
There are, so they saw that some genes that are related to virulence are more produced, basically more expressed when in space, in microgravity. And there are several hypotheses. One is the radiation, but the other is can be also, one thing that is strictly related to that is the fact that when they are in space, they produce generally a thicker biofilm. Biofilm is again that a little film created by microbes that they can form on surfaces. And when they are in these embedded into this thick film, and especially when this film is thicker, they are basically protected to a series of things, and they can become more virulent, they can become more aggressive, they also survive better. Yeah, actually there are several reasons why they increase. Not all of them are known though, but there are quite a number of studies, and the majority of studies saw, well, I'm repeating myself, but they saw that there are some genes that are overexpressed. Sorry, overexpressed is a technical word, but they are more basically produced in space, or basically more used when they are in space. More active, maybe I should do more. Genes that are more active in space.
Okay, thank you for that one. Here's one. This obviously comes from your statement about it comes from Roy and Charlotte Evans, obviously whiskey drinkers who say sherry cask or peated whiskey.
I prefer peated whiskey, but I like also sherry cask.
I know nothing about that.
And you can see there are my bottles of, actually they are not mine. They are my partner bottles, because he is a member of Edinburgh Scottish Malt Society. No, was it? Yeah, Scotch Whiskey Society. So he has all the bottles from, but also books. Please notice also the books.
Yes, I was admiring the books, not the bottles. I was worried about the bottles. One from Billy Russell, talking about biological. Alien life experiments in space. Have you seen the 1971 sci-fi movie, The Andromeda Strain? Is that before your time?
No, I didn't. I have to say I didn't watch enough sci-fi movie apparently, because I remember The Martian and Armageddon, but I didn't. No, I should watch them more, because when I watch them, I also remember Arrival. I really like Arrival because it's quite interesting, but no, I didn't. I will.
Okay, it said that movie had a virulent multi-silicate. No, I can't say anything about that. You're obviously looking at the chat box as well.
Oh, sorry. Yeah.
Yes. So David Dagan says, a brilliant and illuminating lecture on this subject, which should be known about more. And I do hope you can come back with the results of your future research. I think I forecast that from David. One from Alistair. Given the fantastic capabilities of microorganisms on Earth, why have no microorganisms been detected yet on Mars, given the extent to which water has been found near the surface? And the second question from YouTube, do you think it is likely microbes will be found under the Martian surface or poles, or in other bodies such as Europa?
So regarding the first question, is the fact that although microbes are quite amazing, they are not the Martian, the conditions on Mars are very, very hard. For instance, yes, it's been found water under the surface of Mars. It's quite a recent discovery. I think it was from 2008. But in order to be liquid at that conditions, it's probably very, very salty, but salty in nasty elements like chlorine and other chlorine-related species. So it's probably not a very good environment to survive. So who knows, maybe when you go there, you will find that something survived. Most probably, I think, in my opinion, is most probably that we will find that there was something on Mars before, but who knows? Not necessarily still present there, but maybe before, when Mars was a better hospitable environment, there was something there. And regarding yeah, so I don't know if I don't know. It would be amazing if we found microbes under Martian surface. That would be such an amazing result, and especially, and also on Europe, of course. So what I can say, I don't know, but there is hope. The hope is that yes, we'll find something. Although from one point of view, I'm also concerned about that, because I will be kind of sad if then they go and disturb them too much. From one point of view, I don't know. So I will be very excited. I will be very happy, and as an astrobiologist, I will be absolutely amazed. But I also hope that if we find them, we are going to, yes, study them, but still respect them, probably, if from one point of view.
In that case, don't send human beings or anything a human being
Well,
builds.
I still hope that we can be sustainable, that we learn from our mistakes. So,
Alistair has come in with another one. What is the probability, in your opinion, that microbes from space seeded the Earth?
Just as a point, Craig, thank you, Rosa, for answering these questions. They're actually not from me. They're via the stream of YouTube, and I've been sent them by text. I'll just take this moment to thank you, Rosa, for that. The talk was wonderful, and if you could please answer any and all of these questions, that'd be wonderful.
okay. So the third was, what is the probability, in your opinion, that microbes from space seeded the Earth? This is an hypothesis that is called panspermia. I don't know, maybe, in my opinion, well, probably, it's more probably, in my opinion, that maybe organics arrived from space rather than real life. But again, this is an hypothesis. We don't know how lives originated on Earth. So as long as we don't have a final answer, maybe possible, everything may be possible, but I think it may be more the other way around. So as I say, sorry, not the other way around, but that organic compounds arrive from space, maybe, but maybe not even that. Maybe they formed on Earth. And I can't expand further on these three questions. So, okay, that was not a question. I don't know. I'm not sure where to find the other questions, though.
Well, I have another question. This is my own. You mentioned that on this new bioasteroid experiment, you're going to use fungi in addition to microbes. I feel like there's an interesting reason for why you've chosen fungi, but I just wanted to know if there was more details about that you could share with us.
So the fungi that we are going to use are still microorganisms, because they are filamentous unicellular fungi, so not mushrooms, just to understand each other. They are not mushrooms. So they are still microorganisms. They are still microbes. They are, although different types of microbes. So I don't know if you can recall the evolutionary tree of life that it was in the first slide. And there were different branches. Fungi, I call them fungi, sorry, it's probably incorrect, but fungi and bacteria are both microorganisms. Some types of fungi are microorganisms, but from different branches. So the reason to do that is that this time we wanted to check if they were, we wanted to use a different type of microorganisms, so not only bacteria, but also other type of bacteria. And the reason to use that microorganism, that penicillium in particular, is because it's one of the most commonly used microorganisms for biomining on Earth. So we know that it's going to it can perform biomining on Earth, and we hope that it could do the same also in space.
Thank you very much.
I think
You're welcome.
I think we're actually coming to an end. We still have 38 members on the Zoom, but I think we're run out of questions. May I congratulate you? You've lasted to 9:10, which is a long session, Rosa. And I must confess, I am not an astrobiologist. I'm not a biologist by any means, but that was fascinating, absolutely fascinating. And on behalf of the society, I'd like to thank you for coming along, and also to echo what David Dagan says. When you get some more, would you like to come back, please?
Okay, thank you. I will try to make something that is not repetitive, so I need to get together quite a lot of results, not to be repetitive.
We'll give you a couple of weeks to work on the new one.
Okay, thank you. Thank you for inviting me. I'm glad that it was of interest.
No, thank you again. I'm going to close the lecture now, so go and have a refreshment or something, and I'll just speak to the members. Just to reiterate the January meetings on the 14th we have the beginners meeting. The 21st we have Dr. Fabio Biancalana, who's going to go back on and talk about black holes, 100 years of mysteries. So all you black hole people will tune in hopefully on the 21st of January. And I think the astrophotographers will come along on the 28th, but watch this space. Colin is moving the date around a little bit. Expect a newsletter probably by the 7th of January, which should have more details included in it. So keep a watch for that. And remember, if you haven't renewed your subscription, your newsletter will stop. I'm looking at Julian Gibson smiling. So without further ado, Rosa, thank you very much for a lovely lecture, and we will no doubt see you again.
Thank you so much.
Thank
Thank you very
you.
much.